Okay, so this video is going to be over meiosis and particularly the events of meiosis 1. I'm in Foundations of Biology at the University of Iowa, and I just wanted to kind of document all of the slides that my professor posted just to kind of help give you guys a rough idea of what our professor is asking of us to remember. So what I'm going to do is kind of just run through all of this stuff that I have typed up here, and what I'm going to do is just kind of read through it and hopefully just add a couple things here and there. Okay, so meiosis, the DNA is copied, followed by 2 rounds of nuclear segregation, and let's see here, I will just try and follow along with this highlighter here just to help you guys out. The DNA is reduced by 1/2, so that means that each product is unique, and meiosis is found in sex cell division. So I feel like for people to understand this, they need to kind of get a rough idea of what we're looking at. And everybody's seen a human karyotype. It just kind of shows, the 23 pairs of homologous chromosomes. The 2n equals 46. It makes sense. There's 2. So times that by 23, you get 46. 1 through the 22 are autosomes, and the 23rd is going to be your gametes or your sex chromosomes. So let's move on to functions of meiosis. It reduces the chromosomes from diploid to haploid, and it also generates diversity among the daughter cells. Okay. And events of meiosis 1. Duplicated homologous pairs of chromosomes come together and pair along the entire length of the cell. Pairing occurs in prophase 1. This pairing is called synapsis. 4 chromatids of each homologous pair form a tetrad. After pairing, crossover may occur between non-sister chromatids. And let's see here, let me get that out of the way. A chiasm is the exchange of genetic information during prophase 1. So I tried to illustrate this over here. It's just kind of when the chromosomes come into contact with each other, their information kind of switches they trade each other. So that's asthma. And then I just wanted to kind of document all of the phases that go along with meiosis 1. So prophase 1 is the pairing of the homologs. So as you can see here, they are getting together and they're pairing up, and this is also where in prophase is where the genetic information is transferred over from each chromosome. So there are these, I tried to illustrate it here, the chiasmas that I was talking about earlier. And this just states that the DNA was previously replicated in the S phase, which I don't have documented. So we'll move on to metaphase 1. Okay, so this is where the chromosomes or homologs, they will line up along this equator, and the centromes, they will send out these like rope-like structures to attach to them to keep them aligned. In anaphase 1, there's the separation of the homologs to opposite sides of the cell. Pretty self-explanatory. And then to end it off, in meiosis 1, we have telophase 1, which ends up with each nucleus is now a haploid cell.